Experiment 6 is an introduction to the area of qualitative testing, that is, testing for the presence or absence of a certain material. You'll gain practice in using blanks and standards, and identifying unknowns using qualitative tests. The tests that we'll be using in this experiment are the Beilstein test for the presence of halogenated solvents, and the Tollens test for the presence of aldehydes. In this experiment, we'll be using a number of different chemicals. Some of them are test reagents, and some of them are positive or negative standards. We'll also be using a number of unknown samples, but those will just be diluted versions of the standards. While of course each chemical is going to have its own specific hazards associated with it, the reagents that we'll be using in this experiment share a number of similar hazards. Many of them are volatile, some of them are flammable, many are either considered carcinogenic or probable carcinogens, and many of them will cause serious skin or eye damage, or possibly even be absorbed through the skin. Some of them, because they're volatile, can also be inhaled and cause damage to the respiratory tract. And according to the guidelines that we've discussed so far, that means many of these things should be used in a fume hood. Many of the hazards can be mitigated by avoiding contact with the material, so you'll definitely need to be wearing your lab coats and goggles to prevent skin and eye damage. You may also wish to consider wearing gloves. If you do, please know that nitrile gloves can provide protection for all of these chemicals. Now, according to our guidelines, many of these chemicals would require the use of a fume hood because they're both volatile and can cause harmful damage to the respiratory tract. However, the volatility of many of these reagents is fairly low, which means they're not that volatile. And that volatility is further reduced because many of the solutions that we'll be working with are quite dilute. For these reasons, you can work with small amounts of the products on the bench top, so long as you keep them covered whenever they're not in use. The final safety hazard that we'll discuss in this video is the hazardous decomposition of Tollens reagent. If Tollens reagent is left to stand and dries out after use, one of the decomposition products that might form is an explosive material. To prevent this from being an issue, after each lab section, we will deliberately decompose the Tollens reagent with nitric acid to prevent the formation of that hazardous decomposition product. When you're performing a qualitative test, you're looking for the presence or absence of a material. And some of the components that you use in a qualitative test are known as blanks, positive controls, negative controls, and unknowns. A blank is a sample that you run with every qualitative test that's designed to check for contamination. So a blank is prepared with all of the materials that are used in the actual test, except that one chemical that should give you a positive result. So the contents of a blank can be water, any of the test reagents, the glassware that will be used in the rest of the test, and that kind of thing. A positive control is a sample that's used to make sure that the test is working correctly and to confirm what the test results should look like. So a positive control should contain a chemical that you know will give a positive result for the test. A negative control is pretty much the opposite. A negative control contains a chemical that you know should not give a positive result. So you can see what a result will look like if the chemical you're testing for is not present. And finally, the unknowns are samples that contain a chemical that you want to test for the presence or absence of some substance. So, any qualitative test that you run should have 4 samples: a blank, a positive, a negative, and an unknown. And now we'll talk about how to interpret the results of a qualitative test. In this example, we had clear colorless solutions for our blank, positive, negative, and both of our unknown samples. After running the test, some of the solutions have changed color. Our blank has not changed color, and this is pretty much what you would expect. A blank should get no result. There should be 0 change from the blank because it contains no chemicals that should give a positive result. The positive control has changed color. It went from clear and colorless to this bright green color. The change indicates that the test successfully found that whatever substance you were looking for was present in the positive control. The negative control, just like the blank, contained nothing that should get a positive result, and no change was observed. When you look at the unknown 1, again, no change happened, so you can come to one of two conclusions. Either, like the negative control, there's nothing in the unknown sample that should give a positive result. That is, you've identified the absence of your test substance. Or that substance is simply present in such small amounts that you can't identify the presence of it visually. That is, it may have changed color, but the color change is so faint that it's not detectable to the human eye. When you look at unknown 2, you can see that it changed color from clear and colorless to a pale green color. Because it changed color, and it ended up as the same color as your positive control, this indicates the presence of your target substance. Sometimes you're able to take a qualitative test and convert it into a really bad quantitative test. Because the color of the unknown sample was much fainter than that of the positive control, you could conclude that the concentration of your substance in the unknown was less than the concentration in your positive control. For the Beilstein test, you'll need a piece of copper wire, a Bunsen burner, and your samples. Your blank will be distilled water. Your positive control will contain a halogenated solvent. We'll be using dichloromethane. Your negative control will contain a non-halogenated solvent. We'll be using hexanes. And you'll also be testing two unknown samples. To start the test, you should make sure your wire is uncontaminated. You can rinse it with some distilled water, and then place the wire into the flame and hold it there until the color of the flame stabilizes. It'll probably stabilize to an orange color. Once the color is stabilized, remove the wire from the flame and allow it to cool. This will take longer than you expect. After cooling, dip the wire into your sample and make sure that you get a droplet onto the end of the wire. You can then place the wire back into the Bunsen burner flame and observe the flame color. When you're testing the blank, you'll almost certainly see no change in the flame color. After testing the sample, you should rinse the wire off with distilled water, and then you can move on to the next sample. You'd expect the positive control, which contains a halogenated solvent, to turn the flame green. You would expect the negative control, which contains a non-halogenated hexane, to have no change in flame color. You'll then test your unknown samples and record whatever observation you have for flame color. The other test that you'll need to do is the Tollens test for aldehydes. In this test, the blank will be distilled water, the positive control will be formaldehyde, a material that contains an aldehyde. The negative control will be ethanol, and you'll have unknowns to test for the presence or absence of aldehydes. To perform this test, you can obtain a test tube. You'll then add 1 milliliter of Tollens reagent to the test tube. You'll then add 2 drops of the sample to that test tube containing the Tollens reagent and observe what happens. You can then repeat that test for each of the samples. For the blank, you would expect to see no change in appearance from that of the Tollens reagent alone. It should look like a clear and colorless solution. For the formaldehyde positive control, you should see a silver mirror start to form on the inside surface of the test tube. If you're rough with the sample and shake it around, you might not see a silver mirror, but you will see a black precipitate of a very, very fine silver powder. For the ethanol negative control, you should also see no change from the Tollens reagent on its own. Again, it should just look like a clear and colorless solution. And then you'll test your unknown samples and make the appropriate observations for them.